From Microbe TV, this is Immune, episode number 41, recorded on January 22nd, 2021. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, we made it to 2021.
Yeah.
Yay.
And we made it past January 20th too. That's a big date, right?
Ooh, yeah, it's a good time.
Also joining us from
Things are looking better.
From Durham, North Carolina, Steph Langle.
Hey everyone. Glad to be back. Yes, 2021 has brought us surprises and we're still battling SARS-CoV-2. A lot to still discuss, but happy to be here.
And from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. It's also great to be in 2021. And hopefully, things will keep moving in that same direction.
It's looking good for science so far.
Yes.
Our guest today is from across the ocean. She is from Imperial College London. Madina Wane, welcome to Immune.
Hi, thank you so much for having me. I'm really excited to talk about some cool new immunology today.
Great to have you.
Yes. Yeah, we're going to actually be able to discuss some non-SARS-CoV-2 science. I know that we had gotten comments of, there's other things in immunology than SARS-CoV-2, but, that seems to be the hot topic. So it's we'll have fun.
Yeah, it's kind of shocking when you do something else. I feel over on TWIV, we have done nothing but that for over a year. It's really, I get a little sad about that. And same thing here, because there's so much out there, right?
Yeah. Yes.
So we have to slowly move over.
Well, great. Well, Madina, why don't we start off? We'll kick it off by learning more about you. Tell us about, where you're at right now, and kind of some things you're doing in your PhD.
Sure. So I'm just finishing my PhD at Imperial College London, and I submitted my thesis in November. So it's really like the very last stages. Yeah.
Yay, congrats.
Thank you so much. I have my viva on the 10th of February. So, it's really like wrapping up the last stages. It's been really strange to finish the PhD, in the midst of the pandemic. But, part of the thing that's been really helpful is connecting with other scientists and like making virtual connections when, even if you're stuck on a screen all day long writing, at least you can at some point see some people on that screen too. So yeah, so the very kind of final stages and kind of looking at my next steps, hopefully got something in the pipeline, but it's not fully confirmed yet. So I'm not going to share too many details.
You don't want to have a, breaking news on Immune.
Yeah. Well, I'm just waiting until it's all confirmed and then I'll, tell the whole world about it.
So I'm curious, how did the pandemic affect the ability of you to complete your PhD? Because I think we probably have a lot of people listening who are sort of stuck in that same situation where their science is impacted by this.
Yeah, that's a good question. And for me, I think I was quite lucky because I actually finished the kind of like practical side of my research, so all of the experiments and so on, September 2019, actually. Like, time is so weird now that it feels like it was just a few months ago, but it's been, over a year now. So when the pandemic hit, I was already kind of writing my thesis and so on. So luckily for me, it didn't impact it as much. I'd say the biggest impact was just kind of the motivation, like trying to focus on writing whilst everything else in the world was going absolutely haywire. But like I said, luckily I've had kind of support networks in different areas. So that really helped just to kind of push me over the finishing line.
Were you able to go attend meetings? Because one of the big things we do towards the end of PhDs is really start to build that network, and that helps you get postdocs and so forth. Was that challenging for you? Yeah,
so I didn't attend a ton of meetings. I'd done so kind of actually fairly regularly throughout my PhD, which was nice. So I've been able to go to like conferences and workshops throughout my PhD. So I don't feel like I missed out hugely. But I know for a lot of students, like that aspect is something that is really, really having a big impact. But yeah, I guess I'm just lucky that I've been able to like form contacts with people across the, my field and kind of beyond before everything got more complicated. But I think it's also amazing that scientists are adapting to, virtual meetings that can be more accessible in certain ways. So I don't think it's kind of like the end of, all scientific networking. I think it's just adapting to kind of a different style.
So Madina, what drew you to science and what got you into the PhD in immunology? Sort of what was your path to that point?
Yeah, sure. So I guess I have I'd say kind of a boring path into my PhD. I've enjoyed science since I was pretty young. Both my parents are from science backgrounds, so naturally nice. I got quite exposed when I was younger, and like my school was pretty good at my secondary school or high school had some pretty good kind of like science resources and science teaching. So I've always been interested in science, and particularly in like biology. I think the science of kind of living things has always fascinated me. So I did biochemistry for my undergraduate degree. Again, at Imperial. I've been at Imperial for a long time now. But yeah, I did biochemistry and kind of still continued that passion for infectious disease and kind of immunology. I specialized in that for my master's. And obviously now with my PhD, it's focused on immunology. So it's been a pretty linear trajectory. But yeah, I guess my passion for immunology has kind of never wavered over the last few years. So it's been nice to follow that into the PhD.
Where did you grow up?
So I was born in France, but I moved to the UK when I was pretty young. So pretty much most of my schooling has been in the UK. But it's been nice to like have, I've got family like all over the world. So I always have that as like ties to different parts of the world.
For sure. I think that there's something to be said though about, staying in an institution. So you mentioned your undergraduate and then to your PhD. I remember listening to This Week in Virology and Kizzmekia Corbett, who is working on the mRNA Moderna vaccines at NIH. And she mentioned, having done internships at the same place multiple summers. And there's really something to be said about building that breadth of experience. And you're going to find ways to challenge yourself even within the same institution. So it's nice to know, you can have that experience and be successful, but still find ways to kind of, widen your experiences.
Yeah, absolutely.
So Madina, you were involved in organizing Black in Immuno, right? And that was a while ago. Maybe you could give us some of your thoughts on how that went.
Yeah, I'd love to. I guess a few months ago now, you had the other co-founders of Black in Immuno on the podcast, which I really love to listen to as well. I learned a lot about the people that I've been working with for a while, so that was really great. But so Black in Immuno, just maybe for those listening to the podcast who don't know, is we're an organization kind of recently set up over the summer. And basically we wanted to start an initiative that was working to highlight and showcase Black immunologists, because as with a lot of scientific fields, there's really a lack of diversity and a kind of lack of maybe awareness of some of the issues that Black scientists face, like the barriers that they face in progressing in their careers. So for us, being able to do something in our research community, in our field, was really important. And we ran kind of some virtual events and an online kind of social media campaign at the end of November, which went really well. It was on for about a week. And we had we had people kind of tuning in from across the world at different stages of their career and different kind of subfields of immunology. So that was really great. And we actually have the talks that we hosted have been recorded. They're up on YouTube. So if anybody wants to check them out, I definitely recommend. I'm just so inspired listening to all of these talkers, and their enthusiasm for their work really shows. So it's definitely something I think is worth watching.
We could link to that within our show notes. We can provide that link. Great. Yeah, cool.
So that was back in Immuno Week, but we're now kind of building on the organization, and we're setting up the organization as, like a fully recognized nonprofit. And I can talk a bit about some of the work that we're planning, the next steps as well.
Yeah, that'd be great. I mean, you're welcome to chat about that now if you want, or we could wait till towards the end, whatever you'd like.
I can mention it now. So like I mentioned, we're building this nonprofit, and as a result, because we really want to make this initiative sustainable and long lasting, we don't want it to be like something that just trickles away within a year, which I think is actually unfortunately quite common with a lot of advocacy work, because
interesting.
You're relying, especially when you rely on people who, we all have, jobs or
trainees. Trainees.
Yeah, exactly. Like everyone is doing this, on top of everything else that's going on in their lives. So in building a nonprofit and trying to expand our team and building like solid foundations, we're hoping that's something that can continue for a long time. But one of the projects that we're working on next is like an Immuno Hub. So trying to build a platform that will connect up different Black immunologists, for example, using something like a database so people can really, because one of the things that's difficult about being, kind of lone Black scientists in your field or in your department is it's really hard to find other Black scientists. So having something like a database will really help us to build a community, and also hopefully help, non-Black scientists or institutions to find and connect with all these talented individuals. So that's kind of one aspect of the hub. And we're also hoping to use it to just generally provide more support and resources for Black immunologists throughout their career, professional development stuff. We're hoping to work on some longer term programs, maybe around mentorship or, more kind of sustained professional development. So it's something we're working on at the moment. We really want to do it right and do it well and do it with the support of the community. So it's something to look out for.
Yeah, definitely. We'll make sure to include those links and keep interacting. I think that, I attended a lot of the talks and I really enjoyed them, particularly your keynote speaker, Dr. Fox, correct?
Fairfax, Dr. Fairfax, yeah.
Fairfax, yeah. Kiki, right. So, I thought it went very well and you guys did a really great job.
Thank you.
Yeah. Well, excellent. So coming from that, we can kind of talk more about advocacy before we go back to science or just go to the science. But I'm curious, how do you see advocacy fitting within your next step? So within your postdoc, is that something you're looking for in regards to a postdoc lab? Somebody, who would be open to having that be a part of your training? Yeah,
so I think for me, like advocacy is always something that I'm going to want to be involved with. So I definitely look out for, people, institutions, organizations who value that, who value diversity, who recognize issues around that, who will, try and support that work. Because I don't want to have to kind of separate my interests. And one of the really nice things actually about running Black in Immuno was hearing from senior scientists who have been able to continue with their advocacy work during their career and understanding from them like what their challenges were or like where they looked for support from their institutions or, kind of beyond. So Dr. Fairfax in her keynote, that was a big part of her kind of journey and learning how she kind of built her scientific career. So then she could have more of an impact kind of later down the line when she focused a bit more on advocacy. So yeah, that's something I'm keeping in mind for sure.
For sure.
So that's very cool. Right now you're doing all this awesome advocacy in addition to doing a PhD, which is I'm very impressed by. Can you tell us?
I'm very tired.
I understand. Can you tell us a little bit about what you're working on for your PhD?
Yeah, absolutely. So my PhD, so I work my supervisor for my PhD is Maggie Dolman, and in our research group, we focus on using zebrafish as a model for studying different aspects of immunology and inflammation. So actually it's really cool because it's a fairly small group, but everyone kind of works on a slightly different area. But because we all work on it in the context of immunology, we kind of have that like expertise and like overlapping interests. And my research specifically has been focused on respiratory disease. So I know we said that we weren't going to talk about SARS-CoV-2 research only, but like, and I don't work obviously on COVID because it's too new, but, my research is still related to respiratory viral infections. So
when I was reading about you and what you do, I thought, wow, I mean, this is a perfect kind of combination of what's been going on with the pandemic and using different models to study immunology of respiratory infections. Very cool.
So which respiratory infections have you focused on?
Yeah.
So my PhD was basically trying to develop a model, I guess a more general model to study respiratory immunology, or rather evaluate whether zebrafish could be used as a model to study respiratory disease. And I did look at a few respiratory viruses. So for example, I used influenza virus and respiratory syncytial virus, RSV, but really as tools to evaluate whether zebrafish could be used as a model. I also used, synthetic compounds, so things like TLR agonists as well, just to kind of probe the immune response. So actually, I'm really excited about discussing the paper today because it is kind of that extension of developing the model, and it's great preparation for my viva as well. So definitely taking notes. Yeah.
Yeah, actually, your committee contacted us. That's what this is. You're having your viva right now.
Get it out the way. Yeah. Surprise viva.
And for those listening who may not know, I don't know if I would have known in my PhD, but just the terminology. So a viva, correct, is the terminology used at your institution, but is that used commonly in the US?
Oh, maybe this is like a UK, US, kind of translation thing. So I guess it would be called a defense in the US. Is it
defense? Okay.
Right.
Yeah, but the way they're done in the UK is you don't usually have I think in a lot of countries, you basically do like a presentation to an audience, which, in the UK, it's just like you and your examiners usually. So you don't really have that kind of public.
Interesting. Okay.
Yeah, which I think is kind of a shame because I really enjoy sharing my research, and it's kind of nice when you've done work for so long to be able to share it more widely. But
sure.
Anyway, it's just you and your examiners in the UK.
Yeah. Great. Well, just to clarify, because I know terminology, like you mentioned, can be different. So speaking about zebrafish as a model, kind of give our audience, what is a model organism? What is it used for? And for somebody who would ask, why, zebrafish for translational human immunology, how does that work?
Yeah, that's a good question, and something that I've try to address at every stage of my PhD. You always get that question, which is great because we become hopefully better at answering it. But
sure.
I think in general, models, whether they're animals or not, for example, even just studying cells can be is a model itself for studying disease. So it's really just a system that allows you to mimic a certain aspect of the biology that you're interested in. And basically no model is going to perfectly do that. So if you're looking in animals, for example, they're always going to have differences to humans. Even if you're looking at human cells in a dish, they're not going to have the same kind of structure, obviously, and properties as a full, a full person. So really it's about when you're trying to evaluate a model and judge whether it can be useful for the research question you're interested in, you're trying to understand what the properties of that model is, whether it mimics some aspects of the biology that you're interested in, and also what kind of tools there are to be able to study your model and answer your research question.
So for zebrafish, there must be some particular benefits or unique aspects that you can use it for that you can't do, for example, with a mouse. Can you explain what some of those are? Yeah,
absolutely. So I think maybe the most obvious aspect is zebrafish are a lot, well, maybe for people who know zebrafish, but they're a lot smaller than some of the larger animals or mammals that are used in biological research. So they're probably about 3 or 4 centimeters long at adulthood. So they're small, they're cheap, they're really easy to keep. You can buy zebrafish just as like a pet fish. So they're very, I guess, accessible in that sense. And because of that, you can use them in kind of larger quantities or for like more higher throughput work. So that's, I guess, more of the kind of practical economic aspect of it. But in terms of their biology, they're also really useful for investigating what's happening directly inside a living animal. So because they're very small, especially in early stages of development, a lot of people have used them with microscopy techniques to image processes happening live inside the fish, which is a lot harder with larger animals like mice, for example. So it really provides kind of a new window to be able to investigate live physiological processes.
Very cool. And speaking of those processes, I think I saw on your Twitter bio that you more align as a mucosal immunologist, particularly pointing to the gills of the fish as like this model for mucosal immunology. So can you talk a little bit about some of the things you've maybe found in your research about gills, mucosal immunology, and how it relates to translation?
Yeah, of course. So the gills, for those who don't know, are the respiratory tissue of fish. So they don't have lungs, or most fish don't have lungs, but they instead have gills, which perform the kind of gas exchange function in water. And one of the cool things that I've been investigating is, because obviously I'm interested in respiratory viral infection, I'm interested in the immune system, is what kind of immune properties do the gills have? You know, what kind of immune cells do they have? Are they capable of responding to different challenges? So I've been able to look at a variety of different immune cells, and what you find is that the gills are absolutely full of immune cells, mostly lymphocytes, but a whole range of immune cell types, myeloid cell types, lymphoid cell types, and so on. I think that's maybe unsurprising because, like the lungs, the gills are exposed to the external environment. So like all mucosal tissues, they're exposed to the external environment. They have to interact with kind of foreign materials and pathogens and respond in a way that protects the host, but obviously doesn't damage the tissue itself. So one of the things that I investigated in my PhD was whether there was a presence of lymphoid tissue in the gills, because we know that lymphoid tissue is really important for mucosal immunity. So I was able to kind of identify and probe some of the properties of this huge accumulation of lymphocytes in the gills, which no one really understands what its function is. Because, for example, fish don't have lymph nodes as far as we know, and they do have, they have kind of diffuse mucosal associated lymphoid tissue, but they don't have these kind of structured lymph node structures. So I think it's an interesting area to understand almost the evolution of lymphoid tissue, you know, how very early vertebrates, from early vertebrates to, humans, what the process of evolution has been in structures that are supposed to protect us from infection.
That is fascinating. And in fact, after this, I am giving a lecture to my immunology class. And one of the things we're going to be talking about are organs of the immune system. And so now you've given me some excellent facts to tell them about fish not having lymph nodes.
Birds don't have them either.
Awesome. I knew more about birds, but less so about fish. But just a general question about zebrafish as a model. I've always sort of been slightly intimidated when thinking about different types of model systems about the availability of things like antibodies or other resources. Is there good availability of those kind of technical resources in the zebrafish for immunology?
Yeah, it depends what you're interested in. So, for example, if you're interested in kind of gene editing, there's loads of tools. That's one of the things that zebrafish are kind of widely used for. You can use all your CRISPRs and so fairly easily. In terms of antibodies, that's one of the big limitations for zebrafish. I know in mammalian immunology, you can, look at all these like huge panels of markers to investigate like really specific immune cell populations and functions. And unfortunately, because zebrafish is still kind of a young model in the kind of history of biology research, you don't have as many of those tools. So what people have done to kind of overcome that is using transgenic zebrafish lines. So yeah, in contrast, you do have quite a lot of different transgenic zebrafish. So you can investigate different cell populations, different fluorescent proteins and so on, but you're still limited. And to be honest, even our understanding of immune cell markers or kind of immune cell genes is still growing and developing. So even if you did have the antibodies, you still have to characterize those cell populations to understand how they compare to other models.
And do you use much fluorescence in situ hybridizations? Do you do fish?
Yeah, that's something that, like, if you're someone who studies fish and you're trying to like search the literature, there is an annoying number of papers that just come up with like
fish.
Yeah, just fish itself, and you're like, oh, this isn't what I wanted.
This is not what I wanted.
Not the right fish.
But I haven't done it myself, but other like a postdoc in my lab has done fish, so it's definitely possible. Yeah.
Awesome. Well, and you had brought up in some of your work using TLR agonists as a way to study immune responses, and you're a co-author on a paper that looks at a TLR agonist across multiple different species, that kind of comparative immunology. It's very exciting. Could you explain to our audience what a TLR agonist is and how do you use that to study in your model?
Yeah, for sure. So a TLR agonist is a compound that stimulates TLRs. So TLRs are toll-like receptors, and essentially they're one of the type of receptors of kind of in the body that will recognize material from pathogens. So normally they'll recognize a certain type of molecule that's kind of conserved across pathogens. So for example, in my work, I've used a compound that will stimulate TLR 7 and 8. And those TLRs usually recognize RNA, so things like viral RNA. So in terms of my zebrafish research, when I was doing my master's, I started working on a project where we're using this TLR7 or TLR8 agonist to stimulate or to investigate a response in zebrafish gills, but also compare that to responses in mice and in humans. And really it was to kind of, again, to evaluate models and to kind of try and expand the toolkit that respiratory research can use. In humans, for example, you might want to study live virus, but a lot of the time that's not really feasible. So using something like a TLR agonist mimics the sort of pathways that would be activated during a viral infection. So we were able to collaborate with researchers who work on mice and humans, again at Imperial, and compare the responses across the species. And it was pretty interesting to see that all three species responded very similarly in terms of the genes that they expressed and the time of their response to this agonist compound. We used one agonist, called risaquimod. That's the TLR7/8 agonist. We also used a second type of agonist, poly-IC, which stimulates, it mimics double-stranded RNA, and it stimulates a different pathway through TLR3. And what we found when we used that compound is mice did respond, have an immune response, but humans and zebrafish didn't. So for us, it was using those two compounds, we were seeing some conservation of the immune response, but also some differences that indicated actually it's not enough, for example, to just use mice when you're wanting to look at animal models for respiratory disease. You probably need to expand the models that you're using so we can have a more complete picture of the kind of fundamental processes that are happening.
Absolutely. Yeah. And I think that that's an important point that, it's not always about translation. Of course, I know that that's a lot of the end goals for human treatment of human disease, but just the fundamental processes. We talk about that a lot on Immune, and Vincent talks about a lot on TWIV. I mean, you need to know the basic mechanism to be able to build up from that.
Yeah, absolutely.
Excellent. Well, we could now transition to talking about the paper that we've mentioned. Does anybody have any additional questions for Madina? I mean, we can keep peppering her with questions.
I just have one.
About
so
herself as
I'm curious.
Yeah, go.
So I have this view of huge fish tanks, but how do you grow these guys? Are they in little tiny tanks or big tanks or what?
Yeah, no, that's a good question, because to be honest, before I started doing zebrafish research, I also had no idea. So it's a learning process for everyone. But because zebrafish are quite small, you can keep them in fairly small tanks. Obviously, depending on the number of fish you have, you'll have a bigger tank with more fish. But they basically just get put on these big aquarium systems. So you'll have like a whole room filled with many, many tanks and thousands of fish. And because they breed easily as well, so we can breed our fish to kind of get new ones and like look at younger stages and so on. And the like embryos just can be kept in like a little petri dish as well. So yeah, it's pretty easy.
And then when you need some fish
great.
for your
we can move forward with the paper. Madina, we're going to kind of have you lead it if that's fine, and then we will ask questions about it. And we do, and there's a connection here because the lead, not the lead author, but the senior author, we did have on Immune back in February before everything shut down, Irene Salinas. So people can refer back to that episode if they want.
Yeah, she's also one of the co-organizers for Black and Immuno. So it's a very small,
that's great.
Small but mighty world, yeah. But yeah, do you want me to just introduce the paper?
Yeah, that's fine. However much you want to go into it, and then we'll kind of, we'll go back and forth and ask questions, but you can introduce it. That'd be fine.
Sure. So the paper that we're discussing is called A Zebrafish Model for COVID-19 Recapitulates Olfactory and Cardiovascular Pathophysiologies Caused by SARS-CoV-2. So essentially, that paper is trying to investigate the kind of responses, the pathophysiologies that zebrafish have to SARS-CoV-2, to, again, similar to what I've been doing, evaluate whether zebrafish can be used as a model in this area of viral infection research. So the lead author is Aurora Kraus from the University of New Mexico, along with the lead author, Irene Salinas. And there's a few other researchers who I guess they collaborated with from the Pasteur Institute in France and the Paris-Saclay University as well. So got some kind of cross-institute collaboration, which is always nice and actually pretty common in the zebrafish field as well, because we're a small group of people.
Sure. Excellent.
So,
go ahead. Sorry.
Yeah. No, that's all. I'm not sure. Maybe you want to discuss other parts or...
No, that's great. So when they first, so obviously when the COVID-19 pandemic started, I think a lot of us were looking for ways of how we can contribute to learning about this disease, as it was, the forefront of everyone's mind. And of course, many of us were shut down. And so if there was a way we can contribute while the shutdown was happening, that is also of benefit. And so it's interesting here to see, we knew, what Irene does in her model and looking at fish as a way to look at comparative immunology. But this is, in some of the experiments that you can walk us through, looking at how they respond and specific organ systems. So it's not going to recapitulate the entire disease, but what can we learn about specific organ systems that people will recognize as the symptoms that people have when they get this disease?
Yeah, no, that's a really good point. And I think, again, it comes down to understanding that models are not, they can't recapitulate every aspect of the biology of a disease you're interested in. So it's really about focusing on your research question. And in this case, I think this paper was kind of exploring the possibilities, but really focusing, I guess, from their findings on the olfactory tissue. So I guess in humans, that's the kind of tissue that allows you to perceive smells and so on. And obviously with COVID-19, we know that's a kind of really important organ because I think it's something like, I think they said like 40 to 60% of people, lose their sense of smell. It was really interesting at the start of the pandemic how that wasn't, that just wasn't discussed at all. And seeing how that symptom has now become a really kind of a trademark of the infection has been really interesting to see that this kind of science learn and evolve. But so that's one aspect that they focused on. And also with COVID-19, we know that there are kind of issues around cardiopathologies as well. So that's something else that they also kind of touched on in their research.
So I have to ask a question, and I might look stupid right now, but I realized fish had hearts, but I did not realize fish had noses. Am I the only one that didn't realize this? I mean, I have a fish tank and I can see that little mouth, but I mean, is it located the same place? Is it structurally the same? Do they take water in through that? Like how do they sense things this way? Because this is a major part of this paper is using the olfactory organ and determining whether, parts of this virus interfere with that or affect the structure and the survival of those tissues. So can you talk about that a little bit?
Yeah, I mean, your question's completely valid. Like, I don't know why. I think because the way that we see noses, obviously we have a nose that kind of extends from our face. Mm-hmm. So when you don't see that in an animal, you kind of assume that they don't necessarily have that. But zebrafish have, I don't think you can call it a nose, but they have nasal tissue. And their nasal tissue isn't directly connected to their respiratory tissue, which is probably the most obvious difference with humans. Yeah. So the gills and the nasal tissue are kind of completely separate in zebrafish and I guess in other fish species. So which in a way, even though there is that difference, it's an opportunity to study that process in an isolated way. Mm-hmm. So you can just focus on the olfactory tissue without necessarily having to deal with the consequences on respiratory tissue as well, if that makes sense. But obviously that doesn't, then that doesn't recapitulate the aspect of disease that you do see in humans.
Yeah, but that's a good point. I mean, because we know you have cells trafficking cytokines being moved about the body and in this systemic way. And if you do have two organs that are connected, you have that kind of a response in one organ into another. And so this allows you to really separately look at those responses, which is interesting. Great. Now looking at figure 1, I'm curious if you could walk us through why you think it was chosen to use zebrafish larvae to start to look at some of the gene expression differences in response to a particular part of SARS-CoV-2.
Yeah, sure. So for those, again, who don't necessarily know, because, why would you necessarily, but zebrafish larvae are, so they're the youngest stage of zebrafish. Kind of one of the youngest stages of zebrafish, I think up until about 3 days, up until maybe 2 weeks or something is when a zebrafish is considered larvae. So it's kind of the early stage of development. They're extremely small at that stage. They start off being like a few kind of millimeters once they've hatched out of their egg. So it makes them really useful for high throughput studies. So people can use them, for screening of drugs, for screening of toxicology studies and so on. So I think part of developing this model and using larvae is you can, kind of screen different compounds and so on in a very short amount of time. Because zebrafish breed so quickly, you can generate, hundreds and thousands of zebrafish larvae in a short amount of time. So it is important to characterize the response of larvae as well as the older stages of zebrafish, which is slightly more difficult to work.
Sure, for sure. Now looking at what they use, so they did not use the whole virus. They just used a piece of it. So they call it the RBD, the receptor binding domain, to be able to probe. And actually looking at figure 1, it says recombinant spike. I'm sorry, I maybe have jumped ahead. So what do we think about these results? It seems to be only a couple genes were significantly different. From the mucosal immunology side, what did these genes mean to you, Madina?
So the genes that they've looked at ACE2, which is the receptor that binds the spike protein. So naturally, kind of an important gene to look at. They've also looked at some kind of pro-inflammatory cytokine genes, TNF-alpha, IL-1 beta, IL-17, some kind of more chemokine genes, and also interferon, a type 1 interferon Phi1, which is known to be important in antiviral responses in zebrafish, but also in the kind of equivalent genes in humans and mammals as well. So, I mean, it's a kind of a small panel, but I guess later on they look at kind of single cell RNA sequencing and so on. So I guess this is kind of an initial look at some of the kind of key pathways that might be important in the immune response to this kind of viral protein.
Right.
So our listeners may be more familiar with some other types of interferons.
Yeah.
So can you mention how interferon Phi1 might be related and might fit in with some of the other interferons that get a little more attention?
Yeah, sure. So interferon Phi1 is a type 1 interferon. So I guess it's interferon alpha and beta for those who study kind of mammals and so on more. In zebrafish, you have type 1 interferons and you have type 2 interferons. I don't think they haven't identified type 3 interferons, but you have, type 1 and type 2. And one of the important things to remember about zebrafish and fish species generally is because they've had a whole genome duplication event, often you'll get kind of multiple copies of kind of orthologs of mammalian genes. So for interferons, for example, they have I think it's 4 interferon Phi1 genes and so on. So it's always, sometimes it can be a bit of a nightmare, I think, for people working with these kind of model systems that have these duplicated genes, like understanding their function, understanding the kind of homology and so on. But it's just something to bear in mind.
Got it. Madina, I actually need to head out to go teach some things about immunology. I'm fascinated and I would like to stay here and learn, but I will just have to listen to the episode to do that learning. So this was awesome. Thank you so much. It was awesome to meet you.
Yeah, great to meet you too. Good luck teaching.
Thank you, Brianne.
Good luck teaching.
See everybody.
Bye-bye.
Bye,
Brianne.
At any rate, it is a small panel looking at these genes, and maybe why they chose this. And again, we should say this is a preprint. I did interact with Irene about this paper. They're still, working through the peer review process, so there's always that to keep in mind. But it seems to be maybe linking this decrease in interferon, type 1 interferons, is something that is seen in other models, and so it's not the whole virus, but maybe trying to suggest, okay, we can recapitulate some of these differences in this model.
I wonder, Madina, if they make the point in the first section of saying, there is ACE2 in zebrafish, but the contact residues are not conserved between zebrafish ACE2 and, in other words, how the RBD of SARS-CoV-2 would interact with zebra ACE2 probably is not going to happen because the residues aren't conserved. So I wonder if these responses that we see in this paper, the ones that you just talked about, we're going to see effects on heart rate and other effects. They're not likely to involve ACE interaction, right? Are they, in the case of these, is there something else going on? I mean, are there sensors for the protein, for example, that might be mediating these effects rather than ACE2?
Yeah, that's a good question. And it is interesting that they say in that kind of initial analysis that they don't think ACE2 would bind to the zebrafish ACE2 would bind to particularly because they don't use the full spike protein. They only use the kind of binding site of ACE2, of the spike protein. So I guess they don't specify exactly kind of what the sequence of that is, but it's an even shorter form of the protein. So in a way, there's kind of even less opportunity for it to interact with zebrafish cells. I guess they do mention that you have the other receptor, or TMPR.
Empress TMPRSS.
Yeah, that's the one. I'm not sure I don't think they did bioinformatics analysis on it specifically, but there's always a chance that, the protein is interacting with other receptors in zebrafish.
I mean, the one thing I noticed is that there's no control of another protein. Just throw some other protein in and see what happens,
That's right? That's right.
Yeah, I would love to see that as well. I think for the most part they use PBS, so it's a bit, and because as far as I could tell, there wasn't information on kind of how the recombinant protein was produced as well. So it's always hard to know which aspect of your reagent is causing these effects, and what else might be in your kind of in your material.
And so that was what I was going to ask, sort of related to that, is you've done some analysis with viruses and the mucosal response in zebrafish. Were there things that you saw that were similar and some surprises to you in what was up or down regulated in their analysis?
Yeah, actually. So maybe not with figure 1, but later in the paper, they look at the response to whole virus in larvae. Mm-hmm. And they do find kind of increases and differential regulation of type 1 interferons and some more inflammatory, kind of a greater response than you see with just the recombinant spike protein. So for me, that looks kind of similar to the response that you see when you use the TLR7 and 8 agonist that I mentioned previously, where you do have a kind of type 1 immune response signature. You have the interferon response, you have pro-inflammatory cytokine response. And we know that also from like other viral studies in zebrafish and that have used other types of viruses. So it's not entirely surprising that they are getting these kind of responses. Obviously, with the spike protein, it's a bit different, but, as Vincent said, we don't really know what it might be interacting. Whereas with the whole virus, you at least have the genetic material, for example, that we know zebrafish can respond to nucleic acids.
Right.
So I guess if we're looking at this paper as a live view of them developing a model that could recapitulate aspects of the disease, we could maybe say that, having the fish, not the larvae, and giving it the whole virus is going to be maybe a better way to study some of these responses based on what they're seeing here. It's a more broad response and maybe more physiologically relevant.
Yeah, it would be nice to see. I guess maybe that's something that they're going to do next, look at the response in kind of adult fish, because, they could see what the virus does in the olfactory tissue of adult fish rather than just protein.
Right. Well, maybe, but so I have a lot of questions about using the live virus, because it's neat to have fish in BSL-3. But going back to the larva study, so in figure 1, can you talk about some of these, so the average intensity, is that the heartbeat graph? Mm-hmm. Average intensity, yes. Could you talk about what they looked at here and why this may matter for this disease in the context of the model?
Yeah, so basically they looked at the response that larvae had to, again, this recombinant spike protein, and they measured the heart rate. And how that affected the zebrafish larvae. I think it was within fairly short timescales.
Okay. Mm-hmm.
Maybe like 3 hours after administration of the compound. And also just to highlight that this was, I think, continuous exposure to the protein rather than a kind of one-off exposure that they then take away. But again, this goes back to the cardiopathologies that you see in humans infected with COVID-19. So I think obviously there's a lot of research trying to understand why that's the case. That's not something that you see with all respiratory viral infections. So for COVID, I think that's a really kind of key aspect. And one aspect that they looked at in their study, as well as just seeing the effect of the protein, was seeing how whether you could use a drug to basically reverse the effect that the spike protein was having. Again, trying to give some indication of how the zebrafish model could be used.
And again, I think it would be good to have, as you all mentioned, another protein control to know, is this really a response to the RBD in a specific way? Or is this just, you add a protein, to your model system, you're going to get a response. It was interesting that they used an ACE2, not ACE2, but just an ACE inhibitor to try to reverse the difference in average intensity of the heart rates. At the same time, we know that there's not going to be binding to ACE2. So blocking ACE2, I wasn't sure, logically how that works. Didn't know if you had thoughts.
Yeah, it's interesting. And also, I guess I think one of the things, so I'm not super familiar with kind of physiology. So this is still something that I'm kind of learning myself. But
sure.
I know they mentioned, for example, you have ACE2, which does bind spike protein in humans, but you also have ACE, which regulates kind of blood flow and is thought to have an impact on maybe some of the pathologies. So, and I don't really understand how COVID-19 as a virus affects ACE. For example, people have seen kind of decreases, I think, in expression of ACE in response to infection. So it's possible that there are some other factors which influence the kind of physiology, which aren't necessarily linked to ACE2 binding to the spike protein. But I wouldn't be able to kind of guess more than that.
I agree. I think the use of this drug to see if it's dependent on ACE2 binding is probably not right, because the drug the drug affects blood pressure and can reduce heart disease. So it could be that, if the RBD is somehow increasing heart rate by some other mechanism, it would still be tamped down by this drug. So I'm not sure that that's the right drug to use. It's funny because they say in the beginning, this protein probably doesn't bind fish ACE2, and then they say, so it's a little strange.
It is, but it kind of does go back to what I was hearing clinically when, they asked, well, should people stop taking ACE inhibitors? Mm-hmm. Yeah. That was a big question. But again, there's multiple.
And the answer was no, right?
Right, exactly, because it's a different mechanism. There are different ACE receptors, and like Madina mentioned, it probably is impacting physiology in a different way than just direct binding. Yeah.
But the other issue here is that in humans, there are heart issues associated with COVID-19, as you mentioned, and there is a paper which we did on TWIB which indicates that the whole spike protein can affect heart myotubes, and it's the fusogenic activity of it that does that, right? And so but RBD has no fusogenic activity, so I'm not sure what's going on. I really think they need to throw another protein in, maybe maybe you throw BSA in and it makes the heart beat faster. I just don't know, right?
Well, maybe Irene can write us back and see if those are the follow-up experiments that she's doing.
Well, remember, this is a preprint, right? So they may be asked to do these experiments. So
for sure, but it does. These preprints. There's been a lot said about preprints, especially in the COVID era, but at least for our podcast, it's kind of nice because you can walk through and find opportunities to say, you can do this any paper, but this should be done. This needs to be improved upon. It's kind of a working way to learn about scientific processes.
It's really good. I agree for a podcast, but the problem is the press grabs these and they make conclusions which are not quite right all the time, right?
Yes. That is for sure. We've seen that quite a bit in the past year. Well, then I guess moving on to figure 2, and this might be something, Madina, do you get to measure lamina propria width and cilia length in some of your studies because it does involve respiratory function?
So I haven't done any histology myself, but that's something that other people in the lab have done a lot of work on. So I'm not the only one who's worked on gills in the research group that I'm in. We have other people who have worked on the effect of cigarette smoke on gills, for example, and looking at the kind of inflammation and structural damage that occurs to zebrafish gills. So you can characterize the kind of damage that you see, with histology, with different kind of imaging techniques, looking at kind of structural features. Zebrafish are used a lot in kind of regeneration research, kind of tissue repair and regeneration research, because they have an amazing ability to regenerate a lot of their tissues. So I think it's kind of a common approach.
One of the things that just popped in my head, I don't know. I mean, here in the next figure, they're looking at olfactory damage and renal damage caused by this protein. I wonder if the amounts of protein they are using are actually physiologically relevant. Would you ever achieve those levels in an infection, right? I don't think they mentioned that, right?
Yeah, no, that's a good point.
They do say that there is another study that looked at SARS-CoV-2 protein in zebrafish.
Yeah.
And I think in that study they used higher levels of the protein, which led to what they kind of conclude to be higher toxicity. So it seems that at least in this preprint, they're looking at kind of slightly lower amounts, because obviously, if you just get huge damage everywhere, there is also always the concern that it's just some kind of toxic response. Yeah.
Yeah, right. Looking at figures 2 and 3, and I guess this is before we get to some of the single cell data, which you can chat to us about in zebrafish models. But what are some of your thoughts on this? Because I think we are going into, in this figure 4, exposure to food and the olfaction, and how is that measured in zebrafish? Because they don't really have noses.
Yeah, so I tried to look into that because I hadn't really heard of that myself.
Sure.
But I think what they do is they essentially put some kind of probe in the nasal tissue of fish are cavities, so you can access them externally. So I think they must have some amazing system where in their methods they do say they put these kind of probes into the fish to be able to record, I guess, the kind of electrophysiology responses of the fish as they're sensing different types of kind of smells or foods or particles. But it must be a really kind of fragile system because zebrafish is so small and their nasal cavities are also really small. So it must be something that they've, I know kind of this group does look at nasal tissue and nasal immunology a lot. So I guess they probably have that system kind of already in place.
Yeah, I agree. Looking at this, it is funny because it says the nasal flap was removed with fine forceps to expose the olfactory rosettes in a continuous tank water source. So it does seem to be, you have to have good hands to be doing this work.
Yeah, steady hands when you're working with zebrafish for sure.
Yes.
Do they anesthetize them? So you're doing all these little surgical things and injections and exposures while these animals have to stay in the water, right? And then are they? I mean, once you take them out, they can't really breathe, right? And so does that cause actual damage too? And so, what are the limitations in being able to do some of these experiments? Because they say they're introducing, for example, the spike recombinant, the receptor binding domain into these cavities. And as Steph said, there's kind of in the tank. So how does all this really work on the ground, like on the ground, actually holding a tiny little fish in place and then putting this thing back and injecting these things in and
flapping around
or whatever. It seems like you have to immobilize them somehow to be able to do these tiny little surgeries. And I guess you use a dissection scope. Perhaps in a petri dish or something like that.
So I guess I can't comment exactly on the procedures. They have described some of their methods, but it's a bit hard to sometimes understand unless you kind of see it visually.
Yeah.
But I know in my research, so one technique that I've used a lot is applying compounds directly to the gills themselves, rather than, for example, putting it in the water. And it's actually not too difficult. You can anesthetize fish fairly easily. and they're able to stay out of water for some time. So, I would, for example, anesthetize a fish, take it out of the water, and be able to kind of pipette some of my kind of liquid sample onto the gills, and they can stay out for, probably on average, I've kept them out for like 8 minutes or so outside of water. So it's not, and they recover fine from the anesthetic, once you put them back in water. So actually you do have some flexibility in terms of being able to, it's definitely a limitation that you can't keep them out of the water all the time, but it's also an advantage that you can, for example, just put something in the water and expose fish directly, in the way that it's a lot harder to do that through the air for, kind of land-based animals.
Yeah, I always thought that was very cool that you could do mucosal immunization of fish by adding something to the water.
Yeah. That's a big thing in aquaculture, where they want to be able to do that for immunization of fish against diseases that affect aquaculture.
Right. And I remember Vincent bringing up that there are vaccines where you do have to individually vaccinate fish.
Yeah.
That there are technicians, right, who will do this.
Oh, yeah. There are viruses of fish for which we immunize. They have to pick each fish out of the water and give it a shot and throw it back in. Yeah, there are people doing that.
Well, they probably have machines, because they do this for little chicks too.
No, there's no machines. There's people
no
that, for the fish, they're people. Well, we are machines, right?
Wow. Yeah, because for chicks, they just pick them up and they just go doop, and they put them on this little machine that has a little thing that immunizes them, and then they toss them in another bucket and they just pick up and doop, toss them. So I've seen pictures of or videos of how they do that. So yeah, the picking up individual fish is really hard, because you have to get them from one tank to another tank. Yeah, so there's a push. We have people here working on that. It's a push to develop these water-based mucosal vaccine deliveries for fish.
Right.
I think it's also, and the same as in humans, I think it's also because people are interested in mucosal vaccines, because often that is the site where you get infection, or where you first get exposed to a pathogen. So actually, there's a lot of thinking that you might induce a more effective and kind of appropriate response if you're having some kind of mucosal vaccine than, the kind of intramuscular kind of systemic vaccines that we see a lot.
I got to show you a picture of fish being immunized here from Google. See?
Oh my gosh.
There's one, there's another.
Wow, look at that.
Yeah, they're picking them up. And although they do say that you can also immerse them in vaccines for a few minutes, and that can do it as well. So, but
that's cool.
There's a much larger fish than I've ever worked with.
Oh yeah, they're big.
Like this big.
Yeah, I don't think you can pick up a zebrafish with your hands. It's way too small, but we've got other systems.
That's good. Well, I guess before we kind of move into the live virus studies, anything else you'd like to say about the RBD? I mean, I think we could conclude that, we don't really know if it's a specific reaction. We don't have that protein control, but there are decreases in lamina propria width and cilia being a structure really important in respiratory infections. Is cilia and their function something you look at in your research?
I don't, because the gills don't have cilia. So actually that's one of the kind of differences than you see in the lung. So they must have some other system to be able to kind of clear debris and material from the tissue. But I guess what I would say is, even though we don't quite know, what the reason is that they're seeing this kind of damage response, like I mentioned earlier, zebrafish are really great at regenerating their tissue. So actually, then they don't show this in the study, but I would be interested to see how long the damage persists and how long it takes to recover, because we know in mammals, we're really bad at repairing our tissues without like loads of scarring and fibrosis and kind of chronic inflammation. So I think that's one of the benefits of zebrafish. And if we can study some of those pathways and investigate how damage is repaired, then that might be able to give us some clues at how to treat human disease. Yeah,
that's a great point. Yeah, because looking at, they went out to 5 days, looking at the damage, but if you go out further, how long does that take? And then again, with the exposure to food and smells, you see potentially similar reactions, loss of those functions, but they only go out maybe one day. So yeah, that would be very interesting to look at. And then they do some single cell data. Have you worked with single cell data? It's always like beautiful to look at, right? Like I'm looking at a lot of colors, but it can be sometimes difficult to make sense of what am I particularly pulling out of these data?
Yeah, I think I haven't done single cell RNA sequencing myself, just like, there's so many papers now that it's becoming just like a much more accessible and widely used tool, especially actually in zebrafish research, because like I was saying at the beginning, in terms of the kind of immune markers and so on in zebrafish, we don't have a super great understanding of these different markers and different specific cell populations beyond the kind of broad macrophage, T cell, etc. So there's been quite a few zebrafish papers using single cell RNA sequencing to try and map these different populations more precisely. So there's a lot more coming out, but a lot of the data is quite overwhelming, and sometimes it's a bit confusing to understand, like, what are you looking for? Like, what's the research question? Because it's just a lot of information kind of thrown at you. So I think single cell RNA sequencing can be really powerful, but you really have to understand why you're using it in the first place. Otherwise, it's just a lot of data, in my opinion.
Data dump. Yeah,
sure.
That's a great point.
Yeah. Now, what do you think for this group's work, looking at this? Do you have a couple takeaways of the single cell data? I mean, I'm seeing some time changes.
Yeah, to me, that it's a lot more different than I normally see for these types of plots. Like normally you see one population sort of decrease or another population increase or activation states change. But this is really a very different profile of the cell that are there in those different time points for me, at least it looks to me. I don't know what you guys think about it.
Yeah, it's interesting because they mention as well that there are a couple of cell clusters which aren't identified in the control group, but are identified, I think I can't remember what time point, but, in the kind of spike protein exposed group. So you're even getting, whether understanding where those kind of cell clusters come from, if it's changes in their previous cell population or not, or if it's kind of a new cell emerging, especially with the lymphocytes. I think they see a new cluster, and understanding if that's because of a recruitment of cells or whether it's a change in the existing cells is unclear, but there does seem to be quite a lot going on.
And really what we could say about single cell data like these is that getting it out there does allow people a data set to then reference back to, because then, people who are asking maybe those more specific questions, when you don't have anything in the literature to draw from, it can be very challenging then to support maybe what you're seeing. And so I think if anything, maybe there are research questions that they are looking at in this group, but then kind of that public service, I don't know where they'll submit it to, but, assuming you would have to share that would be an open source data set.
Yeah.
Yeah, that's a really great point. And, I know in the research group that I'm in that other people have looked across different data sets, and actually those have been really powerful. Even being able to look between species, for example. So yeah, it's definitely useful, but sometimes I think it's just kind of hard to pass through all of that information in one go.
Sure. So then going on, they did take fish, they put them in BSL-3, and they introduced SARS-CoV-2 in the water, which would be the way you would give fish if you're trying to mimic, I guess, respiratory contact. They did mention that maybe it would be different if they injected them with the virus. What are your thoughts on that in regards to modeling and infection, and the route that you give it in this animal?
Yeah, it's a good question to ask, and it's something actually in my research that I've investigated using different routes of infection. I guess, so when you're exposing a fish to something in the water, even though that does expose the maybe mucosal tissues that you're interested in, so for example, the nasal tissue or the gills, for example, in my case, you also have to remember that you're exposing everything else. You're exposing the gut, you're exposing the skin of the fish. And unlike in humans and in mammals, the skin of fish doesn't have this kind of keratinized surface. So the living cells are kind of exposed beyond the mucous barrier, are quite exposed to the environment. So that's a mucosal tissue in itself, the skin of fish. So by doing that kind of route, it's almost like a kind of systemic challenge. You're just throwing it all to the fish at once. So that's why with my research, I've tried to apply compounds or pathogens to the gills directly to have a slightly more local exposure. And I mean, they've done that with their intranasal exposure, but obviously with larvae, you can't, it's very hard to do that. The most you could do is a local microinjection, I guess. People do that in areas like the hindbrain, but I don't even know if their nasal tissue is developed at that stage. So it might not be feasible.
It's an interesting comparative question, right? Because you're using this as a model to understand SARS-CoV-2 in this case, or in your case, other viral infections. And so if you're trying to understand something similar to a human disease, then maybe doing this specific exposure at a specific place is important to do, because as you said, if you put the fish in the water, put the virus in the water and expose the whole fish, you can infect anywhere. But the flip side of that is, right, if a fish is exposed, it's either going to be exposed because it ate something that has something, or it's in the water, right?
Right.
And so I guess, how do you balance between what's important to understand about fish biology and natural infection routes versus manipulating the model to learn something that maybe we can translate to human? It's an interesting question, because it's something we face with using any animal model that we use.
Yeah, sure.
But I just wanted to sort of hear your thoughts on how you think about that.
Yeah, I guess it comes down to just what you're trying to address. So, I mean, for some people, they do want to look at the kind of more natural infection process, and that might be more important, for example, if you're trying to translate your findings to say, aquaculture. You're interested in fish health specifically. I think sometimes it's useful of thinking about zebrafish as a kind of in-between model. So in between, in vitro research, which is just cells in a dish and very physiologically different, obviously, to a whole organism, and a kind of mammalian system, which maybe is closer anatomically in some respects to humans, but obviously still has its differences. So, yeah, I think with zebrafish, they kind of fit in that in-between space, as well as offering these kind of other advantages.
I think you make a really good point, because I think it's two steps up from tissue culture, right? Tissue culture is mostly, not always, but mostly uniform cell culture, right? Then you have organoids, which can have multiple cell types. And then you have your zebrafish, which is a whole organism, but you might be looking at one system, and then you can just build up from there. I think it's really interesting to think about that, and think about, where you are in the context of being able to look at not just cell responses or cell interactions, but tissue interactions that can provide another layer of information that you couldn't get a different way. I think it's really interesting.
I would have been shocked if SARS-CoV-2 reproduced in zebrafish, frankly. I mean, I suppose they had to do it, but
sure.
I mean, fish are very far from mammals, and so. But they do see decrease of ACE2 and pro-inflammatory cytokine responses. So, frankly, I'm puzzled. I don't get it. I don't know what's going on.
Yeah. Maybe it's the response to the RNA, and so maybe then what we can pull from that is a lot of the responses in our body, our innate immune responses, may not be due to productive viral replication, viral progeny, but the responses, the innate responses to just the RNA being there.
That's why I asked her the question way back when I wasn't so eloquent, but the idea of like what surprised you and what did you see similar? Because a lot of your studies, Madina, were on adding a TLR ligand and looking at the inflammatory response. And so how, especially an mRNA mimic for a TLR7/8. Yeah. So like how similar is this to that in this part of the paper? Because how much of it could just be a TLR response in the absence of any viral replication because there isn't any? Which is interesting.
Yeah. I think that's probably, I mean, to me, that's fairly likely that you are getting some kind of recognition of nucleic acids. And the point about kind of looking at innate immunity, that is something that zebrafish are used to study. That is one aspect of immunology that zebrafish are used a lot for. Actually, partially because in zebrafish larvae, they don't have a developed adaptive immune system. So again, a lot of people use that to isolate the effects of, or the kind of impact of the innate immune system on, say, infection, without the presence of an adaptive immune system. So again, it's kind of, those differences can be used to your advantage depending on, what aspect of biology you're interested in. Even if you're not necessarily going down a translatable route, I think there's still those insights that you can gain from separating different components of the system.
Madina, are there TLR knockouts in zebrafish? I presume so, right?
I actually don't know, but there, like I said, like there's a lot of kind of genetic editing in zebrafish, and I think there's a consortium that's tried to like make loads of different mutants. I think the Sanger Center in the UK, maybe. So there probably is a bunch of TLR knockouts, but a lot of these mutants haven't been characterized as well. So you kind of, a lot of the time you're starting from, not from scratch, but one up from that.
I guess that would be a logical next thing to do here is to look at some TLR knockouts and see if you put virus or RBD in. I mean, in mammals, one of the TLR, what is it, 4? It senses glycoproteins, right?
Two, yeah.
2, TLR2, yeah. So
4 is LPS, yep. Mm-hmm.
So you could do knockouts and see what these responses are caused by,
Yep.
Yeah.
Yep.
Well, great. Well, I think the last part of this paper was just looking at in the live virus challenge fish in the water was, again, differences in fold changes of genes that they had looked at with the larvae. And I guess, Madina, if you had any thoughts on that, but I wanted to kind of bring it back out to another paper that you're co-author on looking at different cytokines, and you highlighted IL-10 as a cytokine that created this homeostasis in the gills of fish. Can you speak to IL-10 and what in your model does homeostasis mean in the gills?
Yeah, sure. So the paper that you're referring to, that was actually a collaboration with some researchers from the University of Manchester. So that was really cool because essentially we had used this system of administering this TLR7/8 agonist to zebrafish and characterizing their inflammatory response. And then these researchers from the University of Manchester have had an IL-10 kind of mutant zebrafish. Actually, that was developed in a different group, but they also had some IL-4. So in zebrafish, the IL-4 kind of homologs or IL-4/13. They have 2, IL-4/13A and IL-4/13B. Anyway, they also had knockouts for those genes. So using the kind of model that we developed with the TLR7 agonist, they were able to investigate, okay, what happens to the response when you knock out these genes? And they found, or I guess we found, that the inflammatory response kind of didn't resolve in the kind of normal timescales when you knock out these genes. So showing how important they are for the regulation homeostasis of the tissue. IL-10 specifically actually is important for just the general homeostasis without any kind of immune challenge. So without it, you do get kind of damage to the gill epithelium and kind of persistent kind of chronic inflammation that just continues without any kind of immune challenge.
And that actually makes me think, is that at all due to T cells or any other autoimmune reactions without the appropriate tamponing of the immune response? Do you know, or is it just all innate?
I would say so. I think when you look at zebrafish larvae, which don't have, remember, functional adaptive immune system,
Okay.
you still get increased inflammation at the earliest stages. So I think there might be some mechanisms affecting T cells, but a lot of it will also just be kind of innate mechanisms as well.
Fair enough. I was thinking of the adult, but yeah, you were talking about in the larvae. Do they have microbiome? They do, right? And so that could be a part of it, right? And so if you are messing with homeostasis, you might be messing with their ability to, hold the microbiome in check.
Yeah, absolutely. Yeah.
Well, great. Well, I guess, we made it to the end of the paper. I think we've learned a lot about zebrafish models and Madina's work. And I guess as we kind of wrap up, Madina, is there anything else you want to talk about or highlight about your work, about I guess your career? What are some things you see yourself doing in the future?
Sure. Big question.
Just the big question. What are you going to do after this?
Or are you going to stick to zebrafish? That's the big question.
That's the question.
So, I won't share too many details because I'm trying to finalize this, but my next step hopefully is going to be working on vaccine research in industry, but finalizing all of that at the moment. But I'm still passionate about immunology. I'm quite interested in being able to develop my kind of knowledge and understanding of other models in translational research. I think working on zebrafish has been a really cool way to understand the tools that immunologists use to try and study immunology. But I guess trying to broaden some of my own understanding beyond the kind of basic biology, kind of classical immunology. I guess now is a great time to go into vaccine research as well. It's always a great time to go into vaccine research, but it's nice that obviously it's the importance of that is recognized kind of more universally as well.
Mm-hmm. Absolutely.
Yeah, I think vaccine research has gotten a boost that's going to last maybe a long time.
Well, we had the immunization and now there's the boost. And so hopefully that will have a more robust response and a longer term response.
Yeah, I love it. Yeah, keep going for sure.
Want to wrap up, Steph?
Yes. Well, we thank you so much for coming on. It was really wonderful to learn about you and we wish you the best. You're a great speaker. You communicate science well, and we hope to just follow your work as you progress.
Oh, thanks so much. I'm excited to see what else the podcast has as well. There's been some great episodes recently, so it's great for me as well.
Yeah. Well, this was wonderful. Thank you.
That's Immune 41. Show notes are at microbe.tv/immune. Send us your questions to immune@microbe.tv. And if you like what we do, consider supporting us. We depend on your support to do this. Microbe.tv/contribute. Our guest, Madina Wane, is at Imperial College London. Thanks, Madina. Pleasure having you.
Thanks so much for having me. Love talking to you all.
Cindy Leifer's at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thanks.
And I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.